The overall goal of this protocol is to improve investigations of trophic ecology, especially from field samples.
This method can help answer key questions in the field of food web ecology, such as the impact of invasive species on trophic interactions. The main advantage is the additional application to more time integrating trophic analyses for the exact same specimens, and that it can be applied by virtually every ecologist. Though we established this approach to provide insight into prey consumption of aquatic consumers, it can also be applied for other purposes, for example to determine the species composition within benthic macroinvertebrate samples. Demonstrating the procedure will be Christian Sodemann, a technician from our laboratory.
To start this experiment, transfer previously dissected predator gastrointestinal tract to a 2 ml SafeLock reaction tube containing 440 microliters of salt extraction buffer. Immediately freeze the samples at -20°C until DNA extraction. After removing samples from the freezer, start DNA extraction by using forceps to add one 5 ml stainless steel bead to each sample tube, and then leave the samples on the bench at room temperature to thaw. Using a bead mill, homogenize the samples for 1 minute at 15 Hz. After spinning down the samples shortly, add 90 microliters of 10% SDS and 5 microliters of 10 mg per milliliter proteinase K to the samples. Next, thoroughly vortex the samples and incubate them at 60°C with constant shaking at 400 rpm on a thermomixer for 1 hour.